In this chapter, we're going to look at movement. And it's kind of a really interesting question, I think. How do we go from thinking about doing something to actually doing it? So in the case of voluntary movement, we start off with this intention, a desire, and we end up physically moving our bodies. And we'll see that it involves integration of numerous brain areas, some that are involved in making decisions about movement, others that are helping to facilitate that movement and make it happen or smooth out the motion. Those neural signals end up in our muscles, and our muscles come in three general varieties: skeletal muscles, also known as striated muscles because they're striped, as you can see here in this picture. Those move the body and the limbs. Smooth muscles are involved in the movements of internal organs, like those used in digestion, and then cardiac muscles, which here in this picture, we're seeing a motor neuron that has left the spinal cord and is innervating these muscle fibers. And we're going to see that in the muscle, these two proteins, myosin and actin, are important in causing a muscle to contract. They react to an action potential in the muscle and allow the muscle to shorten and contract. We'll see that the neurotransmitter involved in what's called the neuromuscular junction is acetylcholine. It depolarizes the muscle fiber and opens calcium channels. And those calcium channels allow calcium to come in and allows myosin to pull on the actin, shortening and contracting the muscle.
Just as it sounds, the neuromuscular junction is a synapse. That is the site where the axon terminal of a somatic neuron innervates a skeletal muscle fiber. Notice that the cells never actually come in contact with one another. The space between the neuron and the muscle fiber is known as the synaptic cleft. The neuron portion of the neuromuscular junction, or NMJ, is called the presynaptic terminal. The muscle portion of the NMJ is called the postsynaptic membrane. An action potential traveling down the axon of the neuron needs to be able to generate an action potential in the membrane of the muscle cell. This happens in 5 major steps. First, an action potential travels down the axon of the neuron and reaches the presynaptic terminal. It causes calcium voltage-gated channels in the presynaptic terminal to open, allowing calcium ions to enter the cell. The influx of calcium allows for vesicles containing an important neurotransmitter known as acetylcholine, or ACH, to be released into the synaptic cleft. Now in the synaptic cleft, the acetylcholine binds to receptors on ligand-gated sodium channels on the postsynaptic membrane. This binding causes the ligand-gated sodium channels to open, and because the sodium concentration in the synaptic cleft is higher than inside the cell, sodium ions rush into the muscle cell. This causes an excitatory postsynaptic potential, or EPSP, in the plasma membrane of the muscle cell. The EPSP causes an action potential to be generated that then travels down the plasma membrane of the muscle cell to ultimately cause muscle contraction. The acetylcholine in the synaptic cleft is broken down by an enzyme known as acetylcholinesterase. Breakdown of acetylcholine is important in order to prevent it from continually causing sodium channels in the postsynaptic membrane to open.
So that video explains what happens at the neuromuscular junction and how the release of acetylcholine triggers an action potential in the muscle fiber, allowing calcium to enter. Now we're going to take a look at how that calcium leads to the muscle contraction.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction.
The calcium that enters the muscle fibers allows the proteins actin and myosin to interact and crawl along each other to contract the muscle.